recording this, so I'm going to say it. So this is your week 2 highlights for what we're doing in our second semester of anatomy and physiology. So we're moving on to the cardio part of cardiovascular. So what we're going to find with this section is, of course, the heart itself is the main pump, the main driving force that is sending the blood around into these different circuits. These circuits are represented by what you see here on the screen. So we have what's called the pulmonary circuit, and then we also have the systemic circuit. Now, in addition, sorry about that. Let me open that up again, a little technical glitch there. So, anyway, in addition to the pulmonary circuit, which goes to and from the lungs, we've got the systemic circuit, which goes to and from the body. And there is a third circuit as well, which is called the coronary or cardiac circuit. But in any case, what we're seeing here is arteries taking blood away from the heart, going to the organs of note, and then veins returning the blood back to the heart. When we look at the heart itself, we've got two sides to it. We've got a right side. Remember, we have to think anatomically, where the right side of the patient as opposed to our own right side. So this is the right side here with deoxygenated blood, left side with oxygenated blood. We have an interventricular and intraatrial septum, which divides the two. So because we are really big homeothermic organisms, we need a very efficient cardiovascular system which separates the oxygenated from the deoxygenated blood. That way we make sure that as this blood is going out over the miles and miles of blood vessels, we have the highest content possible of oxygen to be delivered to the tissues. Now, of course, it's not just respiratory gases. We also have nutrients and ions and hormones and all kinds of things that are being transported in the blood, as we learned in the last section. But in terms of the separation there, that's part of what we're talking about there. There's been some research to show that, again, one of the reasons why we would need a heart like this is that we have all of these different cells, trillions of cells that need oxygen. They are burning ATP to help generate heat to maintain our body temperature. And so because of that, we need a much more efficient heart in that particular way. If you go to cold-blooded organisms like fish and frogs and things like that, they have much simpler hearts. They may have a 2-chamber heart, they may have a 3-chamber heart, etc. But 4-chamber heart works for us quite well. Okay, so the atria, remember, are basically going to be these receiving chambers of the heart, receiving blood back to the heart from the various veins. The ventricles are going to be the powerful chambers of the heart with much thicker walls in order to eject the blood out the arteries and route it to the various circuits. Even in this diagram right here, we can see that the walls of the ventricles are much thicker compared to the walls of the atria. Right? We have relatively thin walls of the atria. All they have to do is drop the blood down, right downstairs to the ventricles. Not too tough. Meanwhile, the ventricles have to eject it out of the heart. And particularly in the left ventricle, it has a much, much thicker wall comparatively, because we're going to be sending that blood through this big systemic circuit. So it needs to be much more powerful. So to be sure, you need to know the aspects of anatomy. So you're going to see that in every single system we come to. You got to learn the different parts, and then that's the anatomy. Then we have to learn the physiology, what those parts do. So you need to know how the heart is oriented, where it sits, the different blood vessels going in and out of it, the different layers and membranes of it. Remember that we've got different types of pericardium, a serous pericardium, a slippery membrane that's on the surface of the heart, as well as lining the cavity of the heart. There's also a fibrous pericardium called the pericardial sac. We have 3 layers to the heart as well. We've got an epicardium on the outside, a myocardium in the middle, and then an endocardium lining the chambers. So these different figures, again, are just going through the different anatomical parts of the heart, both at the surface as well as anterior, posterior, as well as inside of the heart. Remember, you do need to know certain aspects of the coronary circulation. These figures are especially beneficial for lab, so that we can find those particular structures and vessels, etc. And then inside, we see the different chambers. We see the fact that we have 4 different valves. There are muscular ridges in the insides of these chambers, pectinate muscles up here in the atria, trabeculae carinae down here in the ventricles. This just shows, again, that asymmetry between our left and right sides, particularly in the ventricle, where the left ventricle has to be more powerful. Just talking about our different valves, realize they're not all exactly the same. They have different numbers of cuffs to them. The semilunar valves that we see here and the tricuspid valve all had 3 flaps. Meanwhile, the bicuspid valve has 2 flaps. The bicuspid and the tricuspid valves, these atrioventricular valves, also have the chordae tendineae attaching them to the different papillary muscles, which are working kind of like I talk about in lecture as almost like anchors, so that when the ventricles contract and those valves close, to make sure that it's a one-way flow, they don't flap backwards. So it's kind of like, you know, ropes on a hot air balloon and the guys that are anchoring it down. We certainly do get a bit into the cardiac muscle tissue and the cells as well. And you're going to see that again and again in these systems, right? Because it's the cells, which are the smallest living unit, which give rise to the tissue, which gives rise to the organ. So anything that the organ does, it's going to be the cells, the specialized cells, that allow it to do its particular function. So cardiac muscle cells, you'll remember, they are striated due to the overlapping actin and myosin filaments. They're branching, so there's a lot of connectivity to it so that they can actually work as what's called a syncytium. Particularly, there's these intercalated discs that you see here, these little dark lines in between. These intercalated discs have specialized cell junctions, gap junctions in them, that allow a signal, an action potential, to travel directly between cells. So it spreads, if you will, like wildfire, so that all of the cells in a particular area can contract almost simultaneously, working as what we call a syncytium. They're working in sync, even though they're individual cells. Another key aspect that we'll get into that you do need to know is when we talk about excitable cells, we learned about nervous system cells, neurons and their action potentials. We learned about skeletal muscle cells. If you need to review that, go back in your book and check that out. You need to know what ions are responsible for depolarization and repolarization. Is there a hyperpolarization? Go back and review what we learned already about generic action potentials in these other excitable cells and compare them to what we're seeing now with our specialized cardiac muscle cells. Now there's 2 aspects of our cardiac muscle cells that we need to think about, or actually 2 cell types. There are the 99%, the worker cells, kind of like in society, the 99%, which are the cardiac muscle cells that are the contractile cells, making up the myocardium. In addition, there's 1%, the elite, the managers, the bosses. Those ones are called autorhythmic cells. Those ones are found inside of the conduction system of the heart. So in the SA node, in the AV node, in the Purkinje fibers, etc. Okay? So those cells are going to act differently. The autorhythmic cells, as the name indicates, have a natural rhythm to themselves. They generate their own rhythm of action potentials and send those out on a very regular basis. And that's what we're seeing here in this chart. We're seeing the action potentials that are constantly, consistently being formed in these pacemaker cells, or these autorhythmic cells. So you can see there's no resting potential. There's no area of the curve that's flat like we saw before in terms of neurons and things like that. When we look at the ions involved, some of them are ones that we're familiar with. So for instance, we see potassium responsible for repolarization. But when we look at depolarization, in fact, it's calcium. We have calcium channels which are allowing for depolarization. What we call slow channels, which allow for the slow depolarization to threshold, and then fast channels, which open up to actually give us the really speedy depolarization that occurs up towards the top of the curve. So we get this very regular signal that's coming out from these pacemaker cells, these autorhythmic cells. This gets sent out to the worker cell saying, this is when you need to contract. Now, now, now. Now, this pattern that we see here, even though there's not a resting potential, it does have a natural rhythm to it that can be changed. We can speed it up, we can slow it down, etc. But there's no time where it's just going to rest. If you have a heart that's at rest, then effectively you have a heart that's dying. Okay? Okay, so you should know about the autorhythmic cells. These are the different places where you can find them. You should also know about these contractile cells. So if you look at, again, what's going on in the contractile cells, say for instance cells within the ventricles, you can see that their action potential is a bit wacky as well. Okay? This is how you see the action potential here in a contractile cell. So notice it has a very, very fast depolarization like we've seen before. This is due to sodium. And I have to apologize right now, I don't have my stylus, so I'm writing like a third grader and writing sodium like that with my mouse. Okay? So we have depolarization due to sodium, but then notice we have this plateau here at the top that significantly prolongs that action potential. That prolonged depolarization is due to calcium channels again. So calcium channels give us this prolonged depolarization, and then we have repolarization due to potassium channels again. And you can see that in the diagram over to the right, where the permeability, or basically when those different ions are moving, sodium, calcium, and potassium. So you do need to know certainly about the action potentials in these autorhythmic cells and the contractile cells. These are the cells that allow the heart to do what they need to do, so you need to know about their two functions and how they work. So this just goes through that particular aspect. Another aspect or topic having to do with the electrical system or the electrical activity of the heart has to do with the ECG, sometimes called the EKG, but of course in the United States we spell cardio with a C, so technically it is an ECG. But for the ECG, in that repeating set of three waves, the P wave, the QRS wave complex, and the T wave, you need to know what's going on in the heart associated with those three waves. So this is the electrical activity of the heart. The autorhythmic cells and the contractile cells, because they're moving charged particles, that is in essence an electrical signal. And so that electrical signal will move through our conductive tissues and can be measured on the surface of the body. So you need to be able to write out or draw out a typical ECG output with labeling your curves, labeling your axes as well as your curves. So time on the x-axis and then millivolts on the y-axis. Okay? For shame that the book didn't label their y-axis. But you need to know what's going on during each of those curves. Atrial depolarization and repolarization, ventricular depolarization, and then repolarization. This is when the signal is getting to these different chambers, causing the cells to contract and then relax. So we can see that in this figure as well, how that signal is spreading from our conductive cells, our autorhythmic cells, to the contractile cells in these different chambers. And that is then showing up on the ECG output. All right, one of the other things that you'll see as a regular other topic that we will hit each and every system is how is this system coordinated or controlled? How do we change its activity to match the needs of the body and coordinate with other systems? And what you're going to find is there's always going to be a nervous system control, oftentimes autonomic, sympathetic, and parasympathetic. There's going to be an influence of the endocrine system, certain hormones that are going to cause it to speed up and slow down, because those two systems are management systems of the body. They are like puppet masters, making sure everything is coordinated. So we always need to know, okay, how does the autonomic nervous system affect this organ and this organ system? What hormones are involved in controlling and coordinating it? And oftentimes there are aspects of each system that control or coordinate themselves. So for instance, we got autorhythmic cells inside of the heart and the SA node, etc., that even if it was disconnected from the brain and from the endocrine system, it would be able to contract on a regular basis. Okay? But our management systems are going to oversee that, modify it, etc. Okay, other things. You should be able to talk about what the heart sounds are, what's going on inside of what we call the cardiac cycle, when we have this contraction and relaxation going on, systole and diastole, right? Systole, spelled, is systole, contraction of the heart muscle, whether it's in the atria or the ventricles, followed by diastole, the relaxation. both of them are key because the contraction is ejecting the blood, the relaxation is allowing that chamber to fill again, so that on the next contraction cycle we have something to eject. So you need to be able to define and calculate cardiac output, what's going on in the heart based on what is being pushed out in an individual squeeze that we call stroke volume, and then how many of those squeezes we are getting per unit time, and we call that heart rate. That gives us what's called cardiac output. So that gives us an idea about how much blood is being put out by the heart. Okay, so the end of the chapter just kind of goes through again those extrinsic rather than intrinsic factors of control. When you get towards the end, there are also some homeostatic imbalances or diseases and disorders that we talk about. You should know some of the terms that you see here. This just really goes into kind of that coordination and various aspects that can affect cardiac output. Do remember that the heart is part of a circuit, and so anything that happens along the circuit in terms of constricting blood vessels or whatever will affect the heart because it's on that same circuit. So you do see some of that as well. And we do a little bit as well about how the heart develops. Not early embryology or anything, but we do compare fetal structures and how they work to supply the fetal body with blood and do exchanges with the placenta, which is a unique organ, to the fetus, and then what those structures become in the adult and how those things compare. So we really look at that in lab, but it is something that we talk about in lecture as well. Arrhythmias you should know as well, because those are something that you are going to run into in your medical field, so know some of the different categories for this irregular rhythm. And really, that's it for our highlights for the heart. Okay? So besides it being such a sweet organ and allowing us to feel things for each other, etc., it's got a lot of work to do inside of this cardiovascular system and moving the blood around. So you're going to be starting about learning with that. Next, we're going to be learning about the different blood vessels and how they conduct this blood around and work with the heart overall.